Hello, good evening. Okay, I don't know why my image not seeing well. How you guys doing?
Good evening.
Good evening. How's everything? Okay, so I don't know. I'm gonna have to turn the lights on because it seems like my image is kind of dark. Doesn't make a difference, but hey. Oh, something is going on. Okay. So I hope you guys enjoy it last night and today. So we are going back again to these office procedures that medical assistants are usually asked to perform. Any questions before we begin with this class? Okay. So I don't know, my image and it looks like kind of dark. I don't know what it is. Can you guys see me clearly? Of course. Yes, no, maybe. Okay, thank you. All right, so let's continue with this particular chapter. Once again, yesterday we were talking about the appearance or the physical examination of the urine. Remember, we just going to check it out, the urine based on color. Of course, we're gonna check volume as well. So when we get to phlebotomy, I'm gonna show you how to do certain tests with the urine, particularly the chemical examination. Of course, we can do the physical because you're going to identify the colors of the urine. It's not that you're going to be asked on an exam particularly about this, but you might have to do these procedures in the medical office are very common. Of course, doing the physical examination is not something that you're going to be doing much, but the chemical examination, yes, and we will get there. I'm going to explain you how it's done. Okay, so let's go back here to, once again, we still continue with urine. Remember, we were talking about the physical part, which is the appearance, has to be transparent, and the color. Also, we talk about the smell or the odor of the urine. Now we're going to talk about urine output. When we said output means the urine that, we pee out, let's put it that way. So in simple terms, um, the urine that comes out every single time that you go to the bathroom. So that is called urine output. So what causes um, the urine, to be formed? We're not going to talk about exactly what is the urine form, but what is it that, we continuously go to the bathroom? Well, if we have different times going to the bathroom, maybe we peeing a lot, maybe we're not peeing much. So if it is a low urine output, now we're gonna identify the reasons. Low fluid intake. So when we do not drink fluids enough amount or volumes of fluids, like for instance, water, anything that we drink is considered a fluid, of course, but there are some fluids that are not much beneficial for us. But let's say that we drink some fluids. Then the more fluids that you drink, the more urine output you're gonna get, meaning the more times that you're gonna go to the bathroom. And if you do not drink enough fluids, you're not gonna have the urge to go to the bathroom because you don't have enough liquids. So the liquids that you take that are not enough, they're gonna be used, consumed within our cellular system, the tissues. So that is the reason why. Now, that is one of the reasons why you have a low urine output. Remember, you're not drinking enough fluids. The second one, excessive fluid loss. How is that? Well, probably you're doing too much exercise, or you have a physical job that demands, more consumption of the fluids, but you're not replacing them. So that's basically what it is. So you are kind of sweating. So remember, when we sweat, that is fluids coming out from the system, the fluids that we have. So if we do not consume enough, what's gonna happen? We're gonna have low fluid inside our body. So we're gonna be dehydrated. That's basically what it is. And remember, the patients are gonna stop by your office and then gonna tell you that they are kind of tired, that they don't wanna do much. And then you're gonna tell them, well, not you, the medical doctor, is gonna ask several questions. And one of them is gonna be, what type of, fluid intake are they using every day? So if they're not taking enough fluids, well, that is the problem. So that is another thing. And when you ask them to give the sample of urine, so you're gonna have a problem. And then you have to keep them longer, or probably ask them to return to the office in another time and to bring the urine. Or you're gonna ask them to drink fluids and give you the urine. So it's gonna cause a little troublesome right there. Now, what happens when you have high urine output? So remember, we talk about when you do not pee much, what is the reason? And now when you actually pee too often and a lot. So it is the opposite. So you're drinking too much fluids. So remember, there is a medical term in medicine that is called homeostasis. What is homeostasis? Internal balance. Everything inside our body needs to be balanced. Glucose needs to be balanced. You can have more than what is considered the normal ranges, or you can have less than. The same with sodium, potassium, cholesterol. So we have to be balanced. Water is not an exception. Every single chemical nutrient in our body has to be balanced. If you are taking more than you're supposed to be taking, then you're going to create a problem for yourself. And if you're not taking enough, same happens. So either or, you're going to develop some type of problems, conditions. So if you drink too much water, and depends of the activity that you do, you're not consuming the excess of water or liquids or fluids that you are taking. So then those fluids are not going to stay in your body. See, there are several nutrients that do not remain in our body after consumption. So others are retained. Like, for instance, glucose is retained. That's why we develop diabetes. Cholesterol is retained. That is why we develop heart conditions, clotted blood vessels for that reason. Oh. So that's basically how it happens. Oh, I don't know how this move to this place. I was trying to do something, and okay, here, go back again to this. So that's basically what it is. So once again, depends on the activities that you do. You should drink certain amount of fluids. If I have a physical job, then I have to drink more fluids than if I'm teaching. If I'm sitting right here, I'm not doing much. I'm not burning anything. My muscles are not working or using fluids. So therefore, I don't need to drink much water. Because if I drink lots of water and I'm not doing enough exercise or my job is not physical, then I'm going to be going to the bathroom every 5 to 10 minutes. So yes, drinking fluids is good, but all depends of the type of activities that you're doing. So normally you should have at least between 6 to 8 glasses of water per day, or fluids, I might say, because it's not just water, it's everything that contains a liquid. It might be soda, it might be juice, it might be, I don't know, broth with your soup. All this is added to it, to the 6 to 8 glasses of, liquids or fluids per day. Now you have to consider your age, you have to consider your weight, you have to consider your height. So all that plays an important role when you are, deciding to drink this much amount of fluids. This is like medications. If you don't just take medication on your own, because you might intoxicate yourself. You might be overdose, underdose, either or, not good. So you have to go to the doctor, and the medical doctor is going to tell you what is the dose and what is the medication that you should take for this particular condition. The same is water. Water is toxic. If you drink too much water, you're going to get intoxicated. Believe me, I'm not kidding you. So it's not good to drink lots amount of water when it's not needed. Once again, you do an exercise, you practice sports, you do this physical job, and you're sweating, and your muscles are working harder, then you should be drinking more fluids. Other than that, you shouldn't go overboard, because what you do is you are going to remove some of the chemicals from the blood, and you're gonna get it out of the system because you're peeing too often. And the water doesn't come out the same way that you drink it. It goes to the system. It goes to the bloodstream. They passes through the liver, through the kidneys, and then return back. Now we withdraw this urine, and when we get it out, sodium comes in there, potassium comes in there, magnesium comes in there, proteins comes in there, glucose comes in there. So we're going to be removing stuff that probably we needed in our body. Why? Because we're peeing too much. This is like you draining something continuously. It's no longer dirty, but you keep on cleaning it and cleaning it and cleaning it. It's not good. That doesn't work the way. That is one of the major reasons. And many people takes all these ads, these commercials, or just expression that, just drink fluids. Yeah, but you have to drink fluids accordingly. Remember, you have to take in consideration your level of activity, your age, your weight, and your height. Other than that, you're going to end up intoxicated. Believe me, you can actually die for drinking too much fluids. Second problem that causes high urine output, that is chemical. So that is something physical in our body, the reduction of the antidiuretic hormone, or ADH. So in anatomy and physiology, this is covered. This is one of the hormones, once again, that allows the water to be retained in our body. So we have to have some type of reservoir in our body. So when we drink some fluids, this fluid goes through all these processes, goes to the blood. The blood is gonna kind of deliver fluids to every single cell of our body. Whatever is in excess, we're gonna pee it out. Now, if we have a problem because we don't have enough anti-diuretic hormone, that means the hormone that retains water, because the water cannot just leave all at the sudden. So we have to retain a little bit. So continuously is supplying. This is like when we open the faucet. You know you have water there, but you only open the faucet or drink the water when you feel like you're thirsty. That's basically what it is. So these hormones help to retain fluids so they can keep you moving little by little. They start delivering fluids. They start releasing these fluids as long as you need it, as long as you move, you do something. Now, if you don't have enough, this is the problem. You don't have enough this hormone, the ADH, now you're not gonna retain fluids. So the fluids are gonna go out as urine. So you're gonna be boiling all the time. So that's basically what it is. So that is not something that you can control. Probably you heard about people taking medications that are called diuretics. Yes, you probably heard that. Oh, the water pill. Well, this is the opposite of this. So that means that you have too many anti-diuretic hormone. So your body is retaining too much urine. Now you develop edema, which is all this fluid that stays in your body make you look like you're swelling up because you have too many of these. No, you have to have certain amount. If you have too little, then you're gonna be dehydrated. And if you have too much, now you're gonna be floated. That's basically what it is. So for these people that has too many of these anti-diuretic hormone, now we have to give you an diuretic so they can help you release all these fluids that are being retained by these cells, by these hormones. So that's basically what it is. So we have to understand how the body works, because if we do not understand the physiology of all the structures that we have in our body, then, well, we're just going to be like, oh, okay, I'm peeing a lot. So, well, it doesn't matter. I'm not peeing much. Well, it doesn't matter probably. But in reality, something is going on. We are not doing something right, or our body is not functioning properly. That's the reason why we collect your urine, because we need to know whether, the color of the urine, whether the pH of the urine, whether the specific gravity of the urine, whether the volume is normal or not. Third, uncontrolled diabetes. It doesn't have to be uncontrolled. Remember, uncontrolled diabetes means that you are not taking medications for it, or that you're taking medications for it and these medications do not work. So that's basically what it is. So we need to figure it out because this is the medical doctors. So what happens when someone has diabetes? So I will not just say uncontrolled, just having diabetes. One of the symptoms of diabetes is that you're peeing a lot. That is one of the major symptoms. If you pee a lot, that means that you are thirsty all the time. So you want to be drinking something because you're peeing constantly, but your body is saturated with glucose. Think about this. If you eat something that is very sweet, what's going to happen after? You want to drink some fluids. It makes you thirsty. Well, you have lots of glucose running through your blood. So what's going to happen then? Well, you're going to become thirsty. And because you are drinking fluids more than normal, then what is it that you're going to be doing? Again, you're going to be peeing a lot. So those are symptoms. Now, there are some other situations, like, for instance, when you have an urinary tract infection or you're pregnant, then, they make you pee more often. So that is because of the situation that you are in. But if it is a medical condition, has to be treated. Remember, blood, urine, feces, any of the body fluids are evaluated to diagnose the patients. And these are the easiest one. Why? Because the patient can provide it to us, or we can just remove it from the patient like blood, right? So it's a lot easier to negotiate or to, evaluate all these body fluids that are easy to obtain, so to speak. Okay, so we just mentioned urine output. Remember, urine output is very important. Pay attention to this antidiuretic hormone because sometimes they give this on your exam. So what is an antidiuretic? Remember, anti means against. Diuretics is fluids. So antidiuretic hormone means these hormones do not retain urine, do not allow you to pee. They retain the fluids. Diuretics are the opposite. See, diuretic, antidiuretic. Think about antibiotics. What is an antibiotics? Anti against biotics, microorganisms, living organisms, bacteria. So that's basically what it is. So we have hormones that retain fluids in our body for later use. And there are medications that if you retain in too much, then they're going to give you these medications that are going to be acting on these hormones to make it release that excess of fluid. So that's basically what it is. So someone that has problems with excess of fluids, they have kind of swelling, edema. Remember, it's not swelling because you got injured or you have a trauma. No, it is accumulation of fluids within your tissues. That's basically what it is. So then they give you an antidiuretic. Okay, let's move now to the chemical evaluation of urine. There is a very common procedure that we do in the medical office to evaluate the urine, and that is called dipstick. So urine dipstick. So that is the most common test that we do with the urine. So medically speaking, it's called the reagent strip. Why? Because we're going to use certain chemicals and a little strip with a bunch of little colors. Each little color in that particular strip is going to have certain chemical that is going to identify any of these chemicals present in the urine. So when we dip them into the urine, so they're going to change colors. And then we're going to compare those colors with a with one of the major, test strips that are going to be compared with. So that's basically with a model. So that's basically what we're going to do. So it says right here, a reagent strip is a basic diagnosis tool used to determine pathological changes in the patient's urine and standard urinalysis. So whenever the patient comes into the office and the medical doctor asks to collect some urine, we are going to do urinalysis. That's basically what it is. Urinalysis, UA. So anytime that you see that the medical doctor orders UA, you see in a requisition form, and you see this UA immediately you know that this means urinalysis. So that means that you have to ask the patient to give you some sample of urine. Because remember, every single time that the medical doctor orders something, they're gonna tell you, we don't do this test, but by ourselves. Oh, I'm gonna order this. Oh, the patient comes here. So I'm just gonna tell them, no, we can collect the urine in advance, but we do not do any tests until the medical doctor order it. If the medical order, then we do it. If they don't order, we discard the urine. So no problem. Remember that we go ahead and collect the urine from the patient because later on the patient probably is not going to be, ready to give us to give us that particular sample of urine. So for that reason, we collect it in advance when we notice that the patient is complaining about anything with the urinary system or the patient comes for a physical exam. So whenever that happens, usually we order urine for those reasons. So now, once again, as I mentioned before, these reagents are used to cause a chemical reaction or to detect the presence and amount of different substances in the urine sample that we collect from the patient. So we're going to see the reagent strips so you have an idea. Because this test, you don't do it in front of the patient. Most of the time, the patient brings the urine, the patient will send it to the waiting area, or the patient is being evaluated by the medical provider. So we're doing this test over there. The patient doesn't have to be there when we do the test. So probably that's the reason, if you're not familiar with this, is because, when you went to your doctor, you didn't see how they were performing this particular test. So here are the reagent strips in a bottle. So you see that you see a bunch of little squares there. So each one of these is one strip. See, this is one strip, this is one strip, this is one strip. So what we're gonna do, well, my bad. It's not that the way it's done. Just wanted to make things, it goes from here to here. This is one strip, this is one strip, this is one strip. So these are the models that we are going to be comparing. The strip that we're going to remove from here, because here all the strips are going to be here inside these little bottles. So they are coming with a standardized color, which is the first line right here. The first line right here is considered normal. So we're going to compare the strip that we're going to remove from here. We're going to dip it into the patient's urine. Then we're going to compare with this model. And we're going to kind of move the little strip and compare which of these lines they match. So that's basically how it works. So it's very important for us that we understand what is it that we are doing with this. I have a small video, no worries. So you're gonna see how it's done. And when we go back to the school during phlebotomy, I'm going to show you, because part of the phlebotomy is also to cover urine tests. And I'm gonna show you how this test works, because it's a very, very common and easy test to perform. And most of the medical assistants are required to do this particular test. So basically that's how it works. So let's see how this test goes about. So things that you need to remember, we are going to check the expiration date of these bottles. Every single time that you use medications, that you use equipment, that you use any of these supplies that you use in your medical, you need to check expiration date. Remember, when you have your exams, They're going to ask you, we're going to go over, because phlebotomy had those questions. What is the thing that you do before you collect blood in those little tubes? Check the expiration date of the tubes. Here is the same. You have to check the expiration day of these reagent strips, because if you don't do so, the reagent strip has expired, the test is going to be inaccurate. So the test is not going to be valid. It's going to be false positive, or false negative. Either or is bad. False positive is that it's going to tell the test, you're going to read it by error, that the patient has something when actually doesn't. Or the test strip is going to read that the patient is negative when actually the patient is positive. So either or is not good. If the reagent strip tell you that you have something when you don't have, that's not good. Because now I have to medicate you, but I'm going to medicate you for something that you don't have. That's not good. And if you have something and it tells me that you don't have it, now I'm not going to give you the medication. Now you're going to get sicker. Not good, right? So once again, these reagent strips needs to be checked for expiration date. That's very important. There is another thing that you have to check, the lot number. This is like medications. When you document, you have to document the date of the expiration date and the lot number. It is a number that the manufacturer company place on these bottles, but for a period of time that these reagent strips were manufactured. So that's basically what it is. So that's going to tell us from which group of this particular day these bottles were done. Not that is for our concern or the concern of the patient, but it's part of the documentation. Now it says right here, collect a clean catch urine specimen. Now, we went over this with some of you. We talk about clean catch. For some of the procedures that are done with the patient, remember that this is when you give the little urinary cup. You're gonna give 2 toilets to the males patients, and to the females, we're gonna give them 3 toilets. It's like a little, cleaning pads. So they have to clean, they have to wipe themselves before they collect the urine. Then they will draw a little bit of urine into the toilet, and then they collect the rest of the urine in that particular cup. So phlebotomy, we're going to see that during that part. And some other chapters, we have some of this information. So it's very important because this will be questions for exam. So it's going to be a clean catch. Why? Because we're going to clean before we catch it. We clean the genitals, males, females. During phlebotomy, I'm going to explain you how it's done correctly, because it's going to be a question for your national exam. So that's basically what it is. Then immediately after you remove the strip, now you have to recap the bottle immediately after. Why? Because air should not go into that bottle, because it's going to damage or degrade these chemicals that are placed inside each one of these strips on it. So you're going to see it. So that's basically what it is. So very careful when you deal with these strips. You can't just open the bottle and you tend to forget, because the first thing that you want is that you want just go in there and dip it and try to read it. No, cover it up. Because once again, in these bottles, we have like 100 strips. And if you leave it open for a long time, like let's say for 20 minutes, 30 minutes, all these reagent strips inside the bottle are going to be damaged. So you're not going to be able to use it. And if you use it, the results are going to be false positive, false negative. So that's basic. So the results are going to be erroneous. So that's not good. So how do we do the procedure? Once again, we open the bottle, we remove the strip, and then we dip it inside the urinary cup. These little strips are plastic, so they kind of bend. So the bottle doesn't have to be the same length as the strip. You dip the strip and kind of bend it at the bottom and get all the little squares that are colored on that strip wet. They have to be. If you don't do it, then the test is gonna be wrong. It's gonna be erroneous. Then you are going to remove the little strip and you're gonna have a paper towels on your counter and you're just gonna place it on a, on the on the bottom part, not the part that has the little squares, because those are the chemicals there. So you're gonna place it on the other side, or you're just going to kind of blot it or kind of tap it very slowly on the paper towel. You do this only on the sides of the little strip. Why do you do that? To remove excess of urine. That's why you have to dip it and remove it right away. You do not leave it there like, oh, I'm gonna count till 10. No, you dip it and you remove it. And then you kind of remove the excess of urine from the strip, because now you're gonna place the strip against the bottle that contains the model that you have to compare with. Now, if the strip is wet, now you're gonna transfer this urine to the bottle, because you have to place it very close to it, because you're gonna compare each strip with the one that you have to see which of these strips, which of these values matches. So you can document that. It's not easy to say it, but you're gonna be watching the video and you can understand a little better. So that's basically what it is. Always follow the manufacturer's recommendations, guidelines, because that's what you're gonna do. When you're done, you record it in a separate paper, you're writing it down. Remember, you have to wash your hands, you have to wear gloves, because these are body fluids. So you always follow your standard precautions. Then you're gonna discard this little strip on the biohazard waste container, which is a red plastic bag. It's not the Sharps container, it's a red bag. That's basically what it is, because this strip is not a sharp. So that's basically what it is. So here is the reagent strip. Now, if you look right here, they have what is it that we testing for. So here this is leukocytes. So we are checking for white blood cells. Then we are nitrates. Then urobilirubin. Then we're gonna check for proteins. We're gonna check for blood. We're gonna check the pH. We're gonna check the specific gravity. We're gonna check for glucose, for blood. So we're gonna check for a lot of things that are not supposed to be in the urine. And if they are, then now the medical doctor is gonna diagnose you with some type of condition, or they're gonna refer you to a specialist to be evaluated in more detail. So that's basically what it is. So you see, the strips are right here. This first one is normal. So that is how your strip is gonna be, the one that you are going to dip right there in the urine. So it's identical to this one, but after you dip it right there into the urine, every single square right here, if you have some problems, they're going to change the colors. So the colors are not going to be exactly the normal ones if you have something wrong. But if you don't have anything wrong, it's going to match the first strip on the bottle model. So that's basically what it is. And then you're going to put it right here. Oh, no, it doesn't match this one. Then you're going to place it right here close to this one and compare every single of these elements. Then, oh, it doesn't match this. Let's go this one. Or let's go this one. Your strip is going to match one of these. And then you're going to record that. That's basically what you're going to do. It's not difficult. It's just that if you haven't done it, that's, you know, something different. But it's a very, very simple test to do. You know, that's basically what it is. So let's watch a little video right here so you can have a better idea of how this test is performed. So that's basically what we're going to do, okay? Any questions before we play the video? Let me go here and share this video with you. Okay, ready?
Welcome to this ValueMed product demonstration video. We're going to show you how to perform a urine testing strip test, a URS-10. This is a 10 parameter mission urine testing strip used extensively within the NHS and GP surgeries. The format and parameters on this match all the leading global urine testing strips used in healthcare and clinic settings. So to perform a test strip, open the vial. This one's already been sealed, broken. Remove your test strip and seal the vial tightly to protect the remaining URS-10 strips in that vial. Now, when you're ready to do your test, a urine sample needs to be collected, ideally a fresh urine sample to be dipped almost immediately. A urine sample can be kept for up to 24 hours at room temperature, 72 hours in a refrigerator, or the sample can be frozen for up to 28 days and then retested. But the ideal is to test on a fresh urine sample. So when you're ready to do your test, dip the urine test strip completely in the sample. It doesn't have to be a deep sample. The test strip is flexible. Remove it after 3 or 4 seconds and tap it vigorously to remove any excess urine from the test strip. And then you'll see the pads changing. What we're going to do now is start interpreting these at 30 seconds. And at 30 seconds, you read the glucose and bilirubin, the bottom two. You'll see these both match the negative test results, which are in this column on the very left-hand side of the results. At 40 seconds, read your ketone result, again negative. And at 45 seconds, start interpreting the specific gravity. You'll see this isn't entirely normal at 1. The specific gravity, if I go along the row, matches at 1.015 or 1.02, somewhere between those two parameters. Again, that just gives you an indication of the concentration of the urine sample and whether it's within this normal human range. Then at 60 seconds, you're interpreting the blood, pH, protein, urobilinogen, and nitrates. So the almost top 5 pads. Again, these are all negative at 60 seconds. And then the final test strip that you're interpreting is this very top one, which is the test for white cells or leukocytes, which you read at 120 seconds. So you need some form of clock or watch to be able to interpret these accurately. Any colour change that occurs on these lower pads beyond the read times on the vial, you should ignore, and they will continue to react, particularly if I go back and have a look at this blood test result or the pH. You may think that this is starting to look a bit green at a 120 or 200 second read time. That is of no consequence at all. You must interpret the results showing on the pad at the read time specified on the manufacturer's vial. Hope that's been useful to you. Shows you how to perform a quick and easy test. The important thing is to keep the vial completely dry and sealed at all times when you're not using the test strips, to maintain the longevity of the test strips and to read the pads at the correct read time. Thank you for watching.
Okay. So this presentation or this video saying a lot of stuff like you have to be with the clock, with the watch right there, checking seconds.
Hi,
We don't have
I'm
to do
Liam
any
Neeson.
of those things, okay? So that doesn't work that way. So we just do it as fast as we can. We don't have to go through all these minor details. It's just doing it fast. That's basically what it is. Because the colors start changing in every single little square from the reagent strip. That's basically what it is. Any questions? Once again, it's not that you're gonna learn it just by seeing it or by me telling you. You have to do it. And we will practice this during phlebotomy because it's part of phlebotomy class module to go over the urine. So that's basically what it is. Any questions? Anything at all? Okay, so let's continue. So we did, this is the chemical examination of the urine. And for the chemical examination of the urine, we're gonna use, this is the manual part, which is you that you're gonna be performing this, but it also can be done using a analyzer, a little machine there that you're just gonna put some urine on a little a well or a little space, put some drops of urine, and then dip the strip inside the little device, and then it's gonna give you the results digitally. So it's not something that, we all the time gotta do it. Sometimes they just have an analyzer for this, or they just send the urine to the laboratory. So no worries. You have to do it. It's very easy to do it. If you don't have to do it, no problem. You just collect the urine, label it, pack it up, and send it to the laboratory. That is not a big deal. All depends on what is it that your doctor do. Remember, most of these procedures that used to were done very commonly in medical offices no longer are done, but urine usually is. Because remember, these are wave tests. So these are very simple tests that we do in the office and we got the results immediately. So that's basically what it is. Sometimes the medical doctor just want to know if you have an infection or not, or if you have blood in the urine or not, or if you have excess of glucose in your urine or not, or proteins. So we're just going to check it out to determine a fast diagnosis to start you with medication right away. If there are complications or your kidneys or your liver or something else, then, this has to be sent to the laboratory for further evaluation. That cannot be done in the office. So that's basically what it is. See, automated urine analyzer. So instead of you going through all this process or dipping the reagent strip right there or doing the dipstick of urine and asking the patient to collect the urine because you're going to get the urine anyways, instead of that, then you're going to use this instrument or this device to do this particular test. So that's basically what it is. Let's watch the video because this is not something that, once again, you're going to have problems with because the only thing that you have to do is get, the urine from the patient, give proper instructions how to collect the urine. Remember, it is recommended that the patient gets a midstream or clean catch collection of urine because that's the best one. Otherwise, we're going to have false positives because, if you don't clean before you pee, you're going to be drawing all these bacteria that are in the surface of the genitals inside, the urinary cup. And then we're going to read them. So we're going to find something that actually you don't have. So for that reason, the patient has to be prepared prior to it. So to clean themselves. prior to collecting the urine. So let's see how this test is done. This is, once again, a lot easier. So just follow, the prompts, and that's it. Oh, well, I was going to say very simple, and here it is, very simple. So let's review a little bit about what they did right here, because it's very simple. I'm going to talk because he didn't say anything. So this is an automated, urine analyzer for the chemical process of the urine. So it has the screen right here. Now, it didn't say anything, but here we can print out the results. So you do not have to kind of copy from the screen whatever values they give you over there. No, you print it out and you add it to the medical records. That's it. The medical doctor is going to read it and is going to interpret them. You see, this reagent strip has to be dipped completely. Then all these color little squares needs to be wet with urine. Other than that, the color of these squares are not going to change depending on what chemicals the urine is being exposed to it. Now you're going to see something else that probably you didn't understand. This part, it's not that you're measuring anything. What we're doing here is that's exactly what I do. I have a paper towels, so I put it right there by the counter and I tap the little strip on the side, not from the flat part that has the color. Should not be touching anything because it's gonna get contaminated. So you tap just to release the excess of urine. Both sides. I tap this and then I turn around and I tap the old ways. And sometimes on the back, you can kind of pass it, slide it against the paper towels to release, to discard excess of urine. You don't want this machine to be spills with urine. Now you have to clean it, and inside it's very difficult to clean it. So the strip has to be a little dry on the outside, no excess of urine. So that's basically what I wanted to tell you. Not difficult. Once again, as soon as you put the strip right here, that's it. You put it inside. Now his machine is going to start analyzing. Sometimes if it is semi-automatic, you press start. If it is automatic, as soon as the strip goes in, they start immediately evaluating or analyzing that urine. Once again, these machines are going to give you the results and you can print them out, just like a cash register. You just press there and they give you the receipt. So basically, that's how it works. Very simple. A lot easier. You don't have to be reading or matching the color of the reagent strip with the bottle. We just place it right there. But if your office do not have this machine, you have to do it manually. And it's not difficult either. It's just that this is a lot a lot more convenient. Any questions about using this urine analyzer? Because that's basically what we are,
Hello?
Hi, Professor. I can hear you.
Yeah, I'm not the only one. I can't hear nothing.
me? Okay. Can you hear me now? Huh. Hello?
Yeah, we could.
Yes.
Okay, thank you. See, there was something right there when I shut the system unintentionally. Okay, no, okay, so this is the one. Okay, so my bad, guys. So we're talking about the pregnancy test because you didn't hear anything, probably you were reading it. So when we have the pregnancy test, we can do the pregnancy test with urine or blood. Most of the time, we do both. But because urine is so easy to do, but it's not 100% reliable. Well, blood is not 100% reliable either, but it has more specificity. So for that reason, we run urine because it's very easy, because, Females goes to the medical doctor, and the reason why they go to the medical doctor to do the pregnancy test is because they miss their period. Oh, I didn't have my period, so chances are this person might be pregnant. So that's one of the reasons why we do the pregnancy test. So remember, I cannot stress enough that one of the major tips that I can give you is, if you want to be ahead of the game, is to read what the patient is there for, so you know what the medical doctor might be ordering. That's basically what it is. So you are prepared. So remember, the patients do not know what tests the medical doctor are going to be ordering. But because you were there and you know what is the chief complaint of the patient, now you're going to be prepared. If it is the case that the patient says, oh, I want to see the doctor, I miss my period, don't just say, oh, yeah, probably is pregnant. Yeah. No, that's not what we should be thinking only. We should be thinking, wow, I need to collect some urine. So this is the patient that you're going to tell them, excuse me, Miss Smith, this is not Mr. do not have babies. So Miss Smith, if you need to go to the bathroom, use the bathroom, please let me know, okay? So you can collect me some urine. That's it, because you know you're going to use it. So that's how it works. Patient is complaining about burning sensation in urination, or any problem that the patient is peeing too much, not peeing enough. They're talking about voiding, they're talking about urinating, they're talking about urine. So then you already prepare yourself. Pregnancy is the same. Now, the best urine that you can use to detect pregnancy has to be first morning urine, because the first morning urine is a urine that is very concentrated. It's kind of very dark, because at night you're not eating, you're not drinking anything. So, but your body is using the fluids that you got or you drank during the day. So you're gonna have less fluids. So it's gonna be more concentrated. So you're gonna be able to see during this test, this particular hormone. So that's basically what it is. Remember, question for exam. When you do pregnancy test, what are you trying to detect in the urine test? HCG. This is the human chorionic gonadotropin hormone. This is the hormone that is present when you're pregnant. If you're not pregnant, this hormone is not going to be there, because that's the time that the placenta has already formed. So the zygote is already implanted in the placenta. So if you don't have a placenta, you don't have human chorionic gonadotropin hormone, or HCG. So that's basically what it is. So you need to understand this, particularly when you get these questions, regardless whether you have to do the test or not. Remember, things have changed dramatically within time. When I was a medical assistant back in 2000, I did a lot of tests. This was very common test for me. So it was very easy to do it. Nowadays, you know, the patients already get their own, the clients already get their own kit. They go to the pharmacy, they, you know, do it, and they go to the doctor, but still we have to do it, because now we have to do it ourselves or order the blood test. So that's basically what it is. Remember, this test is used to test or measures the amount of this HCG in the blood or in the urine, whatever the case may be. Blood is to confirm that you're actually pregnant. Remember, urine can give us false positives or false negatives. So blood is more specific. So for that reason, after the urine, we gotta send for blood. It's just that the urine is right away. The blood needs to be, you know, analyzed by the laboratory and then waiting for the results. So we need to know to tell the patient what they should follow, what they should do. Because if they are pregnant, they should be avoiding doing certain things that people usually do. Like, for instance, consuming alcohol, drugs, cigarettes, certain medications, because that can, you know, be against, the viability of the fetus that is already developing inside these patients. So that's basically what it is. So that's the pregnancy test, very simple. So all the time, remember, when you do this procedure, you are going to have the little kid. This kid has to be kept in a place that it is not too hot and it is not too cold. We got to check the expiration date of those particular kits for pregnancy. Then we got to get ready a pipette, which is like a dropper. And of course, we're going to ask the patient to bring us the sample of urine. Now we're going to dip the pipette, remove certain amount of urine, place it on the well of the little device, and that's it. Then wait few seconds to see if, the two lines that are there changes colors, particularly the second one, because it's going to be one C for control, T for test. So that's basically what it is. We got to wait at least 3 minutes for that to happen. It's not that you put the drops, oh, it is only one line. Oh, not pregnant. No, wait, because within 3 to 5 minutes, you're going to see the other line, which is the test line, that's going to change colors. So that's basically what it is. So these are the colors, because we're going to see the image later on. How to interpret the test. If it is negative, only a blue line, which is the control line, is going to show up only. Remember, it's going to be two little lines. One says letter C in front, that stands for control. The other one, it is T for test. So that's the most important one. So if you have only one line, it's negative. If you have two lines and the second line is kind of pink or purple, now the test is positive. So that's basically what it is. So let's look at this image right here. So if you never done this test, no worries, this is very simple. So this is the little kit right here, the device that you're going to be using. So now here there are two letters, T for control, T for test. So in this little space right here, when you're going to be putting like 3 to 5 drops of urine, you're going to wait for 3 minutes, and then this line is going to appear because it's the control one that should appear all the time, meaning that the kit works. Then the second one is the T. So in this case, you see only the blue line. If you see only the blue line, this test is negative. Now it's just the test, remember. So the medical doctor is going to decide something else. Same for blood, remember. The second one, this is the positive one. Now you're going to see the two letters again, but the control is there, like in the first sample. But now we're going to see the second line on the test, which is kind of pinkish, reddish, purplish. That's basically what it is. If you see these two lines highlighted, now the presence of the human chorionic gonadotrophic hormone is present. So the patient is assumed to be pregnant. Assumed, not yet confirmed, because we need to send for blood tests. The blood test is going to confirm whether the patient actually is pregnant or not, or the test was wrong done, or the kit was not done properly, or the urine was not collected properly. It was not the first morning urine. Or you are in the first day, so the pregnancy is not showing up because you don't have enough HCG on the urine. So that's basically, remember, these tests are not good if it is within the first 2 weeks of pregnancy, because it's not going to show it. So it's going to be negative. It's going to show like this first one. So, oh no, I'm not pregnant. Good. But it turns out that later on, you end up to be pregnant. So be careful. Now, one of the recommendations that I give to my students is, remember, never tell your patients what the results are. If you do the pregnancy test, the patient is going to be, oh, how is it? How is it? Oh, no, it's negative. No worries. Do not tell them if it is negative. Do not tell them it's positive. Ms. Smith, the doctor is going to see you soon, okay? They will tell you the results. No, but you can see it there, right? Can you read it? Can I see it? The medical doctor is going to be seeing you soon, okay? That's it. You're not revealing anything. There's no way you're going to get in trouble. Because remember, this might be false positive. This might not be the time that, this hormone is present because it's the first days of pregnancy. It's not 100% reliable. So do not tell anything. Lots of situations have happened with the medical assistants. Just go there and tell, the patient results. The patient, oh, I'm not pregnant. So they don't even go to see the doctor. They just leave the office. Now you have a problem. And it's going to be worse if the patient turns out that actually they were pregnant, but you told them that they weren't. So let the doctor decide, let the doctor do everything, let the doctor get in trouble, not you. Remember, that's my recommendation. It's very easy to get in trouble, very easy. But for me, it's a lot harder to get in trouble. You know why? Because I keep my mouth shut. Remember, less is more. It sounds simple. It sounds harmless. Oh, no, it's negative. How do you know that? Just because you see one line on the test? No, doesn't mean anything. Because still I have to go for the blood test, remember, to confirm whether the patient is or not, because these tests, remember, at early age of pregnancy are not reliable. So for that reason, we do not tell the patients, regardless, we do not tell the patient anything about the results. Let the doctor discuss this, and then you will be safe. So that's basically what it is. Okay, any questions? So this is very simple. So here it is a video for you to see it, how they actually perform it, particularly because I have a male student that, might not know how to do it. Females, sometimes they know how to do it at home because now they sell these particular kits. But if you don't know, if you're going to do it, then you're going to learn the proper way to do it. So that's basically what it is. It's, once again, very simple test. Okay, let me go back here. Oh, now I know this. Changing the screen is very close to, the little button that you close the class, that you withdraw the class, and that's basically it. Okay, so here it is.
Pregnancy tests detect human chorionic gonadotropin, or HCG, a hormone produced by the placenta. Before performing a pregnancy test, sanitize your hands. Wear a fluid-impermeable lab coat and disposable gloves. Gather supplies and prepare the testing equipment. Collect the urine specimen. Ideally, use a first morning specimen because it will contain a higher concentration of HCG if the patient is pregnant. Remove the test cassette from the foil pouch. Using the dropper inside the kit, add 3 drops of urine from the specimen to the test cassette. To ensure accurate results, it's critical that the specimen volume be precise. Note the time. You must wait exactly 3 minutes before reading the test results. While you wait, dispose of the dropper, also called a pipette, in a biohazard container. Urine is absorbed through the test and control areas of the test device. Antibodies to HCG within the test cartridge bind to HCG in the urine, causing a color change if the patient is pregnant. If the result is negative, the blue control line appears next to the letter C, but no line is seen next to the letter T, the test. If the result is positive, a blue control line appears next to the letter C, and a pink line appears next to the letter T. If a blue line does not appear in the C area, the test is invalid, and the specimen must be retested using another kit. Discard the cassette in the biohazard container. Remove and discard your gloves. Then, remove your lab coat and sanitize your hands. You must record the results in the patient's record as either positive or negative for pregnancy. Include the date, the time at which the test was read, the date of the patient's last menstrual period, the name of the test, the result, and your signature.
Very simple. See, was a lot better watching the video, I believe. So once again, these tests are very simple to perform, and you will actually learn from someone other tests that are kind of more difficult. But in this case, it's very simple. Okay. Any questions? Okay, let's move on. So we learned already how to do the pregnancy test. Okay, let's talk about now different departments within a laboratory. That's basically what it is. So remember, as the same as we have different departments within the medical, we have different departments within the laboratory. So if you have problems with the stomach, you go to your gastroenterologist. If you have problems with your heart blood vessels, you go to your cardiologist, right? If you have problems with your joints, your articulation, you go to the rheumatologist. If you have problems with your teeth, you go to the dentist, right? So these are different practices. These are different specialties. But now within the laboratory, we're also gonna have different departments. So if you send the blood for certain particular evaluations, analysis, the blood is not gonna go just to this particular department. No, there are different departments that processes those body fluids. So that's basically what it is. So now you're gonna analyze the blood. You're gonna see if red blood cells, white blood cells, platelets, all this is normal or abnormal. Well, that you send it to hematology, because hematology is the study of blood. And what is blood? For those who took anatomy and physiology with me, you should know by now that hematology is the study of blood, and blood is made of two components: the liquid portion, which is called plasma, and the solid part, which is red blood cells, white blood cells, and platelets. So we are going to evaluate these components. So that's basically what it is. Then we have serology. Do not worry about that. We will discuss that in phlebotomy, because you're not gonna understand it. It's not simple, because that's a process after removing the blood and placing it in some tubes that have certain chemicals. So that's basically what it is. Now, blood chemistry. Blood chemistry is every particular chemical that we have in our body. So we have tons of chemicals in our body. They're needed there. Or sometimes that they are not needed, they are toxic. So we're gonna find them in the blood. So let's say, for instance, we're gonna do chemistry. What is part of chemistry? Oh, cholesterol, glucose. Remember, hematology is just for the components of the blood. Here is chemicals, sodium, potassium, magnesium, all of the periodic table elements, whatever we have in our body. So you're gonna find the blood chemistry. So we are going to draw blood to do this. We usually don't do many wave tests in the office, except for cholesterol, glucose, hematocrit, probably something that we can do right there, right away to see what the patient has. But other than that, we send to the laboratory. Microbiology. Microbiology, as the word implies, is to analyze microbes, microorganisms. So if we suspect that the patient has an infection with these microorganisms like bacteria, viral infections, fungal infections, or parasites, we're going to send it to microbiology. And of course, there is another test that is not here because it's part of the laboratory, but I don't see it here, is pathology. Why? Because pathology is when we send these samples of tissue, biopsies, to be analyzed for possible cause of some diseases like cancer, or the reason why, the patient died, or to determine the DNA of a particular patient or person that dies, and we don't know who that is. So that's basically what it is. So think about this because it's very important. When we get to phlebotomy, we're going to talk about microbiology because they're going to ask you a question there. Hematology and microbiology. So it will be more details when we go to phlebotomy. But once again, if they want to analyze anything, any infection that you probably have in your blood, then we're going to do microbiology. Remember, because these are microorganisms. If they're going to evaluate blood, the components of blood, red blood cells, white blood cells, platelets, and plasma, then we are going to do hematology. Chemistry is a very common test. Every single time that you come to see the doctor, they're gonna order chemistry and hematology. Those tests, we are going to do it. That's a routine test because we need to know how your body's doing, how your chemicals are doing. Are they balanced or unbalanced, below or above normal ranges? So that's basically what we have right here. So we have to be very, very careful, whenever we see these questions in our exam, because all of these sounds like the same, but actually in real life, it's not the same. So we're going to pay attention to all the details because there is a reason why we have different departments. Because every single time that you send this particular body fluids or sample to the laboratory is one. Of course, this is like a mall, but it has different department stores within. So the same is the laboratory. We're going to send it to the laboratory, but the laboratory is going to deliver it to the pertaining or corresponding department so they can do the analysis. Because here, the first one, hematology, is going to be evaluated. The medical doctor that works there is a hematologist. So that's basically what it is. Chemistry is a chemist. It's a medical that specializes in all these chemicals. It's not the same that is going to be evaluating everything. No, each one does just like the medical specialist. If you have problems with your heart, you're going to go to the cardiologist. You're not going to go to the neurologist. So each department is going to be processing, analyzing, and giving you the results of whatever of these tests that you need to be analyzed. And microbiology, once again, anything that has to do with microorganisms. So bacteria, viruses, fungi, and parasites. So keep it in mind. So, as we go through all these modules, and we kind of repeating some of the stuff over and over, it's going to stick in your brains. So you can understand that that's how it works. So hematology, as I mentioned before, the study of blood. So what is it that we're going to be seeing there? Morphological appearance of blood cells. Morphological means the shape. Each cell in our body has a different shape. Red blood cells have a different shape from platelets and white blood cells. And white blood cells, we have 5 types of white blood cells. Each one looks different, and they have different functions, of course. White blood cells, defense is the defense mechanism. The red blood cells, we're going to see later on, they are the one that carries oxygen and remove carbon dioxide. So that's how it works. And the platelets, these are the ones that works to clot the blood. And whenever we have more than, normal ranges or less than normal ranges, now we know what's the problem with you. Not that we know what is the problem, but now we know how to address your situation. So we're going to send for more evaluations, more tests, maybe to see a specialist to determine what is the problem that you have. What happened? Okay. Oh, I'm still there. Okay. So laboratory analysis remember, because hematology is a very, very common test that we do, because we're going to analyze red blood cells. And within the red blood cells, we have hemoglobin. We have. So this particular analysis of blood is very, very important, because that's where we are going to find out or detect some medical conditions. It says pathological conditions. Pathological is disease that you may have. So every single time that your patient comes to the office, the medical doctor is going to order CBC. CBC is complete blood count. So that is right here, blood cell count. So that's what we're seeing. It's not that we're going to count them, but we're going to analyze the blood, and the analyzer, the machines, these automatic machines, are going to count how many red blood cells you have, how many white blood cells, how many platelets, and if they are normal, abnormal, above, below normal ranges. So that's basically what it is. So they're going to determine what's the problem that you have. If it's not, then, they're going to order some other evaluations, some treatments, medications, for whatever they find. So that's what's important. So CBC, particularly with differential, because it's when we count all the red blood cells, white blood cells, platelets, and to determine what's the problem. So that's basically what it is. So it's a more detailed process of, analyzing blood. Okay, so here it is. So when we do CBC, remember, complete blood count, this is what we are going to evaluate or analyze. Now we have to collect certain amount of blood, because this is done for that purpose. When we get to phlebotomy, we're going to talk about, what is the color of the tube and what is the order that the tube needs to be collected and what is the chemicals inside and what's the name of those chemicals that are going to preserve, protect the blood, so to speak. So that's basically what it is. Some of the components within the blood, remember, hemoglobin, hematocrit. Now the reticulocytes, these are nothing else than, immature red blood cells. When the red blood cells are babies, we call it reticulocytes. Why? Because sometimes you can have a normal amount of red blood cells, but they are immature. Immature red blood cells are not good. So they have certain time that they have to become mature. And if they don't become mature, then they don't have no functions, because the only ones that functions are the adult ones. Now, for those who already took phlebotomy and we took anatomy and physiology, we talk about that, these red blood cells last 120 days. So I do a blood test and I see that you have normal amount of red blood cells, but then you are anemic. So then I said, oh, wait a minute. How can that be? Yeah, possible. Why? Because you don't have mature red blood cells. You have more immature than mature red blood cells. So think about the children. What is the children do at home? What is it that they help with supporting the household? Not at all. We, the adults, we need to respond for them. So the same here. The immature red blood cells are not good because only the mature red blood cells contains hemoglobin. Immature red blood cells do not contain hemoglobin. And hemoglobin is the protein that carries oxygen from the lungs to the blood to every single cell of our body. Now, if we don't have hemoglobin, but we have red blood cells, but no use. Because if they don't have hemoglobin, only adult red blood cells contains hemoglobin. So that's basically what it is. So it's very important for us to understand, the physiology, the morphology of the cells to see if they are normal or abnormal. So when we evaluate the cells, or they are normal, just have something that is called sickle cell, which is, a deformity of the cell, the red blood cells. And if they are deformed, they are damaged. If they are damaged, they don't carry hemoglobin. Now you develop sickle cell. See, a red blood cell is round, like a donut shape, and it has a little space right here. Here is what hemoglobin is at, right here at the center. If you have sickle cell, this is the cell that you have. Oh, what is the center? What is the hemoglobin? Right here, doesn't exist. If you have reticulocytes, now you have a very small cell, but it doesn't have hemoglobin. So you have to have an adult red blood cells in order for you to have hemoglobin and have enough red blood cells. Other than that, you are anemic. Now they have to find out what type of anemia you have, or what type of damage you have to the cells, so you can do the appropriate treatment for the patient. So that's basically what it is. So once again, it's very important for us to understand anatomy and physiology. I'm giving you a little information about these cells because some of you have not gone through anatomy and physiology. So I'm walking you through, but we're going to have a chapter when we're going to talk about this. 2-3 chapters. So we're talking about here, we're going to talk about phlebotomy, and we are going to talk about anatomy and physiology. So kind of, going to move around with these definitions so you can retain them, so you can understand what's going on. It says right here, remember CBC stands for complete blood count. Don't forget this because these are questions for exam. CBC, complete blood count, complete blood count, complete blood count. This is one of the most common tests that is run as a routine test. This is like, you know, a standard, gold standard for medical offices. Blood tests, you're gonna get complete blood count, CBC, and you're gonna get chemistry. That's it. And if you have something else, then you add other tubes. But these are the routine ones, even though if you don't have any complaints, you just go there for physical exam, we run this test. Because we need to know how your blood count is it and how your chemistry is it. Remember, sometimes, you don't have signs and symptoms, so you don't complain. But then something is boiling up inside your body. And whenever you have the signs and symptoms, sometimes it's going to be a little too late for the treatment. But if we will cut it on time, now we can kind of, solve the problem a lot easier. So that's basically what it is. So let's move to hemoglobin. Remember that hemoglobin is one of those components of the red blood cells. So that's basically what it is. And one of the reasons that we do this is because we are going to make sure that the patient is not anemic. Remember, one of the most common blood conditions is anemia. Anemia. And what is anemia? Low hemoglobin levels. And why low hemoglobin levels affect us? Because hemoglobin is the one that carries oxygen to every single cell of our body. And without oxygen, we can't survive. Without oxygen, the cells die or the cells becomes weak. Think about this if you ever felt like, oh my God, I don't want to do anything. I'm so lazy. I feel like I don't want to do anything at all. I want just kind of sleeping. I want to be sleeping all the time. So probably you have anemia because you feel exhausted, you feel tired, you start doing a little something. Oh, no, I can't do it because you don't have enough oxygen. Oxygen is the fuel that we need so we can be active. But also hemoglobin removes carbon dioxide, CO2, which is the waste product of oxygen. After the oxygen has been used, carbon dioxide is the byproduct, is the waste. So that's basically what it is. So we need it for a lot of reasons. Think about it. Without oxygen, we can survive. One of the most common types of anemia is iron deficiency anemia. So keep in mind, I highlighted right there on the first line. So remember, anemia, iron deficiency anemia is one of the most common because that has to do with diet, the things that you eat, whether they contain iron or not. So if you have a question in your exam and they ask you, which of the following is the most common types of anemia? Iron deficiency. What is the type of anemia that you develop due to food intake or diet? Oh, iron deficiency anemia, because this is the one that they like to ask, because it's the most common. So that's basically what it is. So how's the test performed? It can be done in two different ways. It can be just by pricking or sticking your finger, doing finger stick, or by drawing blood on one of these CBC tubes, which is the purple color tube. Once again, we will discuss that when we get to phlebotomy. For now, you can just do the finger stick, remove couple of drops from there. That's it. We're gonna put it in a little device, a little strip, and that's it. We put a drop of blood in a little strip and place it inside a glucometer, and that's it, or a hemoglobin device, and that's it. We're gonna get that particular results. So that's basically. Or once again, we can get blood because we are doing other tests. So what I'm gonna be pricking or pinching your fingers if I know that I have blood from your vein in a tube. So I'm just gonna remove a little bit from there and do the test. You just need 1 or 2 drops, 1 drop. So that's basically what it is. There is a small device that is approved by CLIA, which is the Clinical Laboratory Improvement Amendment, which is the organization that deals with medical offices tests. So that is called the HemoCube. HemoCube is the little analyzer. It's a little machine when we're gonna place the strip with, the blood that has been collected already, and the machine is gonna do the reading, the analysis, and it's gonna give us the results. There is no other way. We don't have like the urine, the dipstick. Oh, I'm gonna place it right here and I'm gonna try. No, it might be through a slide, a different test, but here gram stain. But who's gonna do that? No. So that is very, complex, and we don't do it. Medical doctors do it, but not anymore. That is something that is done now by the laboratory. So that's basically what it is. So remember, you're gonna be having questions when they're gonna say, okay, so what type of tests you as a medical assistant can run at the POL? What is POL? Physician's Office Laboratory. Remember, in these laboratories, they are actually, well, they are not actually laboratories. They just give them the name because that's when you collect specimens, but you actually do not analyze them over there. You have to send it to an external laboratory or to the laboratory if you are in a hospital, because doctors' offices do not have a laboratory, a real laboratory that processes and analyzes these blood samples. So that's basically what it is. But this is a very simple test using a HemoCube because it's considered CLIA test. These are wave tests. Later on, we're going to see again wave tests. Any test that you, as the medical assistant or the nurse, evaluates or analyze in the doctor's office with a very small equipment, very not inexpensive equipment. It is called wave test that is approved by CLIA. This is the Clinical Laboratory Improvement Amendment. So if you have a question when they ask you, okay, the procedures that you do at the doctor's office are approved by who? The Clinical Laboratory Improvement Amendment, which is called CLIA. So that's basically what it is. So remember, POL stands for Physician Office Laboratory. This is not a real laboratory, because the real laboratory is where they analyze every single specimen. Here, we just collect them and we send them. But the ones that we are allowed to process over there are the ones that are approved by CLIA, that are called wave tests, because they are not presenting any risks. So that's basically what it is. Okay, so here it is. See, CLIA wave hemoglobin analyzer. So the urine, the dipstick, is approved by CLIA. Cholesterol levels that we do in the office, and we give the results because we don't need specialized people to do these tests. Anybody can do it. We don't need the special equipment to do it. And then to the patient doing the test. If the pregnancy test came negative or positive, well, patient is not gonna have any problems. If your glucose levels are this or that, well, you're not gonna cause any harm to the patient. So that's basically what it is. So in this case, remember, every single test that is done in the doctor's office, quote unquote, laboratory, are CLIA wave tests. Any test that you do in the office and you get the results right there because it's easy to process, you don't need a qualified person to do the test because the test is going to reveal by itself. The results are going to be there. So basically, that is called a CLIA wave test. Now, the results are kind of accurate and reliable. That's what we do it. But they are not difficult tests. And the benefit or the advantage of doing these small tests in the office is because the medical provider is going to have the opportunity to evaluate the results and to actually diagnose the patient right there in the office, or make the appropriate referrals or other more, more detailed exams for the patient. So the only thing that we're gonna do right here is we're gonna use your finger. Most of the time, you're just gonna do finger stick if hemoglobin is the only test. But if I have collected already as a medical assistant of phlebotomist, I have collected already blood for CBC, I just remove a little bit from there with a pipette or with a syringe. That's it. Very simple. Let's talk about hemoglobin levels or hemoglobin standards. What are the readings? What is considered normal? What are the ranges? We're going to talk about only men and women, males and females. Why? Because in children, everything changes. Because they are still growing, still developing. So it's very difficult. And for the children, they're not going to ask you. So the normal values or ranges of hemoglobin for males are from 14 to 18 grams per deciliters. G/dL means grams per deciliter. For females, it's 12 to 16 grams per deciliters. The ranges are different because female has less red blood cells than males. That's why the ranges in males are higher, because males have more red blood cells. So therefore, hemoglobin should be higher. So that's basically what it is. Now, these ranges are not asked like this. When we get to phlebotomy, we're going to talk about what is the question about the ranges of hemoglobin. What can be considered anemia? So that's basically what it is. For now, we're going to leave it here like this. I'm not going to go any in depth with this. Just know that, hemoglobin levels are, for males, once again, 14 to 18, for females, 12 to 16. Now, what happens if you have more than 18 grams per deciliter? It's not good. What about if you have less than 14? Not good. What about females? Less than 12? Not good. More than 16? Not good. Remember, here doesn't apply less is more or more is better. No. When it comes to balance, homeostasis, remember, homeostasis, internal balance. Everything in our body has to be balanced. everything. Glucose has to be in certain ranges, cholesterol, certain ranges, potassium, magnesium, chloride, iron, everything has to be certain ranges. Above these ranges, not good. Below these ranges, not good. That's when we know that you have problems, you have deficiency or excess of something. And you shouldn't have deficiency, you shouldn't have excess. It should be between the normal ranges if you are not sick, if you are kind of healthy, so to speak. So that's basically what it is. Everything has to be balanced in our body. Remember, including water. Drinking too much, not good. Not drinking enough, not good. So that's basically what it is. We covered this already. So how do we go about doing the process of collecting the sample for doing the test of to test for hemoglobin? So the first thing that we got to do, once again, we're going to identify our patient. We're going to introduce ourselves. This got to be for everything that we do. We're going to wash our hands. We're going to wear gloves. We're going to get the equipment that we need, because that's what it is. Once again, we will discuss this in more details, doing actually the procedure when we do phlebotomy, because we got to do finger stick as well. So you're going to find the suitable finger. The fingers that are only used to do the finger stick are the third and the fourth finger, the middle and the annular finger. Only these two fingers. You don't use the thumb, you don't use the index, you don't use the pinky. Those are not acceptable. We will discuss this in phlebotomy. So after you get the finger of your choice, whether it's the third or the fourth finger, you're gonna wipe the finger, you're gonna clean it with alcohol, you're gonna use an appropriate lancet. If it is an adult or if it is a child, you have to use the appropriate length of the lancet, which you're gonna pinch or poke the finger. As soon as you pinch the finger, that first drop of blood needs to be wiped out with a gauze. You don't use the first drop of blood for any test. No test that you do, you need to include the first drop of blood. Why? Because it's contaminated. We're going to remove that, and then we're going to squeeze the finger again, get a second drop of blood, place it right there on the strip of the test strip, and then we're going to place it on the automated machine. So that's basically how it is. Very simple. Remember, the first drop of blood after you do finger stick needs to be wiped out. That drop is not used. We go about the second, the third, whatever else, depending on the test. Otherwise, we're gonna get the wrong results. That drop of blood is contaminated. So you're gonna place the cuvette, which is the little tray where you're gonna be placing, the drop of blood, or the strip, because there are different models. And then you're gonna get the volume, because these are automated machines. That's it. You're gonna once again document results, not tell the patient what the results are. You have to discard the equipment. Whatever you don't need, the lancet, you have to place it in the charts container. The cuvette, because it's a little, tray, you might use it as a chart container as well. Follow the policies of your organization. or the manufacturer of the instrument that you're using. So that's basically what it is. It is not difficult, just like when we did, the urine through the analyzer, the urine analyzer, or other tests that can be done in that fashion using an automated machine. So that's basically how it works. Let's kind of watch a short video right here. I believe that's how the test is done, but I will show you how the test is done when we get to the bottom. It's here using okay. I don't know where this is going. Okay, let me share you a video.
Welcome to the HemoQ HB301 system instructions on how to collect a capillary sample and perform a measurement. In order to obtain reliable and accurate results, correct capillary sampling collection technique is key. To perform a test, the cuvette holder should be in its loading position. The display will show 3 flashing dashes and the HemoQ symbol. Make sure the patient's hand is warm and relaxed. Use the middle or ring finger for sampling. Avoid fingers with rings on. Clean the fingertip with disinfectant and let it dry. Stimulate the blood flow by applying a light pressure from the top of the knuckle towards the fingertip. Keep a light pressure to puncture the side of the fingertip using a lancet. Wipe away the first 2 to 3 drops. Then press lightly until another drop appears. Fill the micro cuvette. Make sure the drop of blood is large enough to fill it completely. Do not refill. Wipe off excess blood from the outside of the cuvette. Make a visual inspection of the micro cuvette. If there are bubbles or if the micro cuvette is not completely filled with blood, discard and fill another one. Place the micro cuvette in the cuvette holder and start measurement by gently pushing the holder to its measuring position. The result is displayed within 3 seconds. The result is displayed as long as the cuvette holder is in measuring position. Discard the micro cuvette after measurement. For your own safety, please remember to wear protective gloves. Handle blood with care, and follow local safety procedures for disposal of used micro cuvettes. For more information, please visit.
Okay, so that is one of the videos. Now, the person who is narrating the video says, oh, wipe off the first, second, and third. You don't need to do that. It's just the first, okay? Some of the devices or the manufacturer company from the device, maybe that's the recommendation for them. But for us, according to CLIA, you don't have to do that. So that's basically what it is. So no worries. It's just the first drop, not the second, the third, the first drop only. That's how we are going to do it. I think that I have another video right here that shows exactly that. Check all the equipment that is used. Sharps container. Very simple. So when you have these devices that work, it's a lot more practical to do the test. So again, you're gonna do whatever you have available over there. If you have to do it otherwise or not to do it, send it to the laboratory. That's exactly what you're gonna do. Okay, so let's go back here to the PowerPoint presentation. And so we're done with this HemoCute test or hemoglobin test. That's basically how it is. Now we're going to take a break, and when we come back, we're going to go over other very simple tests that are done in the doctor's office or POL, remember, physician's office laboratory. Okay, so we are going to return to class 7:40, and we're going to continue with this chapter. Enjoy your break. See you later. Okay, so see you later.
Okay, thank you.
You're welcome. Enjoy your break.
to make a distinction that's safe to enter. We normally look for a fully habitable structure that includes
Okay, so we are back.
Here, teacher.
Okay, so let's continue with this chapter and hopefully we're gonna finish it up today. Let me get it. Okay. So great. So question for exam before we give you definitions about hematocrit. So hematocrit, once again, remember hemoglobin, hemoglobin, hematocrit, hema, blood. Remember, this has to do with complete blood count or hematology. That's basically how we understand this. Now, hematocrit, what is the purpose of checking for hematocrit?
But they're incredibly important for the health of this river.
Hello? Somebody is asking me something? Okay, so we can use hematocrit also to make sure that, the patient is not anemic. That's another thing, depending if it has lower levels, because what we're gonna measure is packed red cells. So in other words, what we are trying to find out is how fast these red blood cells goes all the way down to the test tubes. So when we process the blood in the office or in the laboratory, either or putting the test tubes inside a centrifuge. So after we place it in a centrifuge, we're gonna wait until, the blood, all the solids, and the solids are, remember, red blood cells, white blood cells, and platelets, they go all the way to the bottom. So that's basically what it is. It's gonna measure the percentage of packed blood cells in the value of whole blood. So that's basically what it is. So we're gonna see if you have enough red blood cells. This is a different test, but it's very similar to hemoglobin, but it's more complex to perform this test because we have to different issue. The other one, we use an analyzer. So a little device then is just going to give you the results. Here we have to process the blood. Instead of just getting a drop of blood, here we have to collect blood and place it in this. And that's when we're going to see the separation of the liquids and the solids in its particular layers. So that's basically what it is. Now, again, in general, you don't have to do it because when we order CBC, all these tests are analyzed in the laboratory. So we don't have to do, we don't do hematocrit in the office generally. We do hemoglobin, but we don't do hematocrit because it's a little more complex. We rather send this to the laboratory and the laboratory is going to process it and is going to send us the results. So that's basically what it is. So let's look at the test tube. This is the tube. Remember, this is a very, very tiny tube. So we use it also collecting blood from the finger, and we start putting the drops right there at one of the ends of this tube. through this place blood. So you place the finger around here, and the drops are gonna go right here. And then this tube, this very tiny tube, is put in a centrifuge. But this is a different centrifuge from regular blood. You have to lay them down like this. They are going horizontal on a plate that has little groups. So we put in these groups, we put the little tubes. And then we centrifuge this. So we turn this on and start rotating, and the blood is going to separate in between, what is the liquids and the solids, which is white blood cells, platelets, and the red blood cells. So that's basically what it is. We're going to see all these layers right here. See, this is the plasma. Here is the Buffy coat, which is white blood cells and platelets. And here we have the red blood cells. So that's how it works. Now, this is not anything. This is clay, because we secure this little tube with clay so the blood doesn't seep out. So that's basically what it is. These little tubes, I have them. I'm going to show it to you when we have phlebotomy, by the time that we cover phlebotomy. So I'm going to show you the little tubes, not that we're going to do the test, because we don't have the machine for that. But yes, I'm going to show you the little tubes that are very, very tiny and very fragile. They can actually break very easily, and sometimes you don't even see them. They're very, very small. So that's basically what it is. One of the reasons that you have this particular topic right here is not because you're going to be doing the test, probably not. Chances, like 95% chances that you won't. Depends where you're working at, because this is nowadays done in a laboratory, analyzed by the external laboratory, but because you have questions on your exam. So remember, the question on the exam is, what is it that, what we're measuring with the hematocrit percentage? Is the percentage opaque blood cells, meaning red blood cells, white blood cells, and platelets. It's going to separate in different layers. So the machine is going to analyze how many of red blood cells, how many of white blood cells, how many platelets do we have in this particular sample of blood. That's basically what it is. Remember, this is with blood. This is hematology. So one of the diagnoses that we can get through this, because it's red blood cells, is if the patient is anemic. So hemoglobin can tell us, is the most common test for diagnosing hemoglobin. But sometimes we have to go a little farther and do hematocrit as well. Now, if you have higher number of red blood cells, so that is a condition that is called polycythemia. It's actually polycythemia vera, B-E-R-A. Poly means many, cy means cells, emia means blood. So there are many many cells in the blood. So you have too many. You have more than normal red blood cells in your sample of blood. So for that reason, this person needs to go and get a treatment, because that's not good. It's like not having enough red blood cells, you need treatment. If you have too many, you need treatment. You have to remove them. So that's basically how it works. So for that reason, we go over this particular topic because you are going to be asked during the test, remember. So which of the following tests, blood tests, measures the percentage of packed red blood cells, hematocrit, or vice versa? Hematocrit, what is hematocrit? What is the test? Oh, it's a test to measure the percentage of packed red blood cells. That's it. Just remember percentage. No other test measure the percentage of packed blood cells. Hemoglobin doesn't measure that. So that's basically what it is. So that's one of the key points or tips that I'm giving you to answer this. And remember, it's going to separate, the blood in 3 layers after it's being processed or centrifuged. Plasma, the buffy coat, and the Pac cells. Pac cells are just red blood cells. The buffy coat is white blood cells and platelets. And the plasma is the liquid portion of blood after being centrifuged. Centrifuge means that this blood is being placed in a machine that spin these tubes, or particularly these microtubes. And when it's spin, then it's like, using a blender. You're gonna place your fruits right there and it's gonna liquefy. And the solids are gonna go to the bottom and the liquid part is gonna stay on top. So that's basically what it is, what happens with blood when it's processed. Whether it's for hematocrit or any other test, blood always is being centrifuged and it separates in the same 3 layers, regardless. But hematocrit is a little different procedure because we use different tubes. And once again, something that probably not going to be doing. So this is the testing. So here are the hematocrit levels. You're not going to be asked about hematocrit levels at all. Hemoglobin levels, yes, but hematocrit, no. But you will learn what exactly is the question about hemoglobin, not exactly what is the normal ranges for male and for females, but during phlebotomy, I'm going to tell you what is the test that is going to be done to detect anemia, which is hemoglobin, but what will be the values that will be considered anemia. So that's basically what it is. So we will discuss that when we get to phlebotomy once again. So here are some values of hematocrit. Men, 40%, remember 42% to 52%, because we are measuring the percentage. See, here it is, the percentage sign. So that's how you're gonna remember. Hemoglobin is grams per deciliters. You don't see percentages, but you're not gonna have numbers there that you're gonna see the percentage. You're gonna have definitions only. So for that reason, just please recall this. For females, 37 to 47%. Remember, always think about females have less red blood cells. It's not something that are made up. It's our nature. That's how we are, made of. More red blood cells for males, less red blood cells for females. That's how it is. For children, newborns, infants, they don't ask this question because it varies. It will be very unfair for a test to ask you about children. Anyone underneath 18 years of age, it will be very unfair because you're still in the process of developing. So every single year, the values are changing. So we don't actually being tested for this. Not for blood pressure, not for the pulse, not for anything. We do all this, but we just record then whatever values we get. But we do not ask during exams, particularly for these programs. If you are a medical doctor, yes, then you have to know all the values because you have to diagnose the patient and treat the patient. So you need to know what are the ranges of normal, whether it's below or above normal ranges. So that is a different issue. But in your program, we don't focus much on that. Just is a lot simpler. Differential blood cell count. So here, let me explain you what is this, because I said it before, but let's go over again. Usually you have two tests in the form, the order that the medical doctor is going to give you when you are asked to draw blood. That's called a requisition form. Remember, you don't do tests without the doctor giving you an order. It's not a verbal order. It's not like, hey, you collect this, and then you have to write it down. Oh my God, what I'm going to write down? No. It's going to be a form, and you're going to be given that, and it's marked every single test that you are going to be collecting. Once again, these requisition forms we will go over when we get into phlebotomy. So the doctor, for anything, if you have to give an injection, you have to do collection of urine, they have to give you an order. Remember, we do not do it by ourselves. I don't decide upon myself to do a test for this. Oh, I'm going to order this for this test. No, we can collect urine just to make sure that the patient doesn't go to the bathroom, and then we're going to have problems trying to get the urine from the patient because the patient already went to the bathroom. That's a different issue. And if the urine was needed, fine. If it was not needed, well, what the heck, we just discard it. But we do not order, we'll start doing this test now, and I'm going to give the results to the doctor before they order. No, we don't do that. So when the doctor gives you the order, there are two tests that we can do. One is CBC, just like that, CBC, complete blood count, which is going to tell me how many red blood cells do I have, how many white blood cells, how many platelets. That's it. But if I tell you CBC with differential, that changes the whole dynamics. CBC with, this means with differential, diff. So usually you will see it like this. So this is differential. So when we do a blood count with differential, so be careful, because when the doctor orders CBC, check it out. Is it CBC with differential or just CBC? The good thing here is that it's the same tube. It's the same amount of blood. But the tests that we're going to evaluate with the differential are going to be more detailed. So that's basically what it is. So we are going to see red blood cells, not just red blood cells. Here we're going to see all the components of red blood cells: hematocrit, hemoglobin, sedimentation rate, erythrocytes. So there are more things versus CBC is just in general. How many red blood cells? No more. Not what it is within the red blood cells. So now we're going to start kind of being more detailed. So that is differential. Now, white blood cells. White blood cells, if you have CBC alone, standing alone test, it's going to be just how many white blood cells. But if you order it with differential, now it's going to be white blood cells. There are 4 types. How many leukocytes? How many neutrophils? How many eosinophils? How many basophils? How many lymphocytes? Because there are 5 groups. Other than that, they're just going to give me a round number. Here is each one of the components of the red blood cells, the white blood cells. Platelets do not have components. Platelets are just platelets. But then we have the other ones. So that's basically what it is. Now, for these tests, they can be ordered and done directly in, analyzed by a laboratory, or it can be done by a blood smear. This is done by doctors. That is it. What we do is we prepare the slide, the little glass slide with smear. That's we're going to prepare it. Then the medical doctor is going to use a microscope to look through the specimen. and, count these particular cells. So that is a very, very complex test. So for that reason, a medical doctor has to do it. Recent doctors, they're not doing that. I never did that. I did that when I was in the university, just, part of the practicals. But I never did this test practicing medicine. Everything we send it to the laboratory because it's a very tedious test and sometimes might not be accurate. So you send it to the experts. Some of the very old medical doctors back in the days, where, they didn't have other choice than, they were in an isolated kind of medical center and they didn't have a laboratory there to do these tests. Well, they have to do it. They learn how to do it. I learned how to do it, but it's very difficult to interpret this. So to be honest with you, I never did it when I was practicing. Number one, I work in emergency. So most of the things, I didn't do much of these very small kind of tests. No, if it is an emergency, we send it right there to the hospital because this is done mostly in doctor's offices. But I didn't work in a doctor's office. I work in emergency in a hospital. We have the laboratory right there, a real laboratory. So every single test that we order for anything, any part of body fluids, we send it to the laboratory. We were, focused more on saving the patient lives versus reading a test. So this will be the blood smear. So once again, this is not done anymore. In doctor's offices, no. We don't get paid for it much. So for that reason, we send it to the laboratory. So we get paid the same amount of money as if we would have done it because we have to give the results. But the laboratory is going to get paid. But if we do it in the office, we spend like an hour, 2 hours trying to do it. And at the end of the day, we're not going to get paid for that. So we don't do it. Not anymore. Insurances don't pay for that. So we send it to the lab, pay to the lab. They send me the results, so I don't have to go through all these hassles. So that's basically what it is. So what we're going to do is we're going to have a slide like this. See? There is a slide where we're going to place the drop of blood. And we're going to have another slide that is called the spreader. So we are going to do a smear. That's basically what it is. So it is actually to do this smear is a little troublesome. I had to try like over 100 times to get it right. So again, thankfully, I had to do it because this is not done anymore in the offices. So, but you're going to have questions from this for your exam. So once again, you're going to place a drop of blood or 2 drops of blood right there into the slide. We're going to, you're going to make sure that the blood is close to the, let me go right here. So the drop of blood is around here that is going to be placed because here is where you're going to have this frosted part of the slide when you're going to place the patient name, day of birth, today's day, all the patient's information got to go here and it's going to be written with pencil, not pen, pencil, because this is glass and it's a frosted. So in this part, only this part where you're going to have the label, or nowadays, you have computerized labels, you just place the label of the patient in this part. So the drop is going to be just there touching the frosted part. Then you're going to place the slide right here. The angle has to be between 30 to 45 because that's going to be the question for your exam, 30 degree to 45 degree. That's how inclined this glass, this is the angle right here. Oh, I'm sorry, the angle is right here. Otherwise, it will be like a right angle, and that's not. is right here. This is the angle, has to be between, you know, the angle right here that forms the slider, and this slide has to be this. So that's basically what it is. And then you kind of run this, see the arrow right here, you run this, the blood right here, when you place the drop, the blood spreads around the edge of the slider. And then you run, you push this, slide this, and the blood smear is gonna be here. So that's basically what it is. So we have it right here. This is how it's supposed to be. It's supposed to be very, very faint smear. It should not have bubbles there. It should not be thick, because then it's not gonna work. So that's basically what it is. So questions for the exam. Now, before we get there, it's right here. Here it is. With the pencil label, patient's name, the frosted, see, I told you already, on the frosted part of the slide, in one of the ends, you're gonna write down the patient's name, day of birth, remember, 2 points identification, the day that the slide was done. That is it, nothing else. Once again, you can do it by removing from the figure, or if you have done venipuncture, collecting it from a lavender color tap tube, which is the correct tube for hematology, then you're gonna transfer from the tube the drops, or you're just gonna place the drops from your finger over there, either or. So once again, this test is not much done anymore. Then you're gonna do the smear. Place a glass slider or spreader directly in front of the drop of the blood at 30 to 45 angle degree. This is one question for the exam. What is the angle of the spreader during a peripheral blood smear? 30 to 45. It's not 90, it's not 100, it's not 20, it's not 15, it's 30 to 45 degree. If you don't see 30 to 45, it is 30, it's gotta be there. If you don't see 30, it's gotta be 45. So you have choices. One of the options is gonna say 30 degree. If you don't see this, then you're gonna see 45 degree. Either or 30 to 45. I haven't been told that, you know, that's the one. Usually 45 or 30 degree. That's the answer of the degree of the angle when you are performing a peripheral blood smear. This is blood smear, remember. Now, the second question for your exam. The way that the smear has to be, it has to look like a feather. See how the feather looks at the end? Like this. So it looks like this. So that is basically how it's going to look, like a feather edge. And that is an answer for one of the questions of your exam. So when you are preparing the peripheral blood smear, how do they have to look? Feather edge. That's it. It's like feather. So once again, if you look here, it looks like a feather that is fading at the end. So this has to be very, very thin layer. No spaces, no bubbles, nothing. It has to be flat, no thickness, because it's not going to be able to be read. So, or evaluated, analyze, can do that. So that's basically how it is. So once again, these testing organizations, they do not evolve with time. So things have changed a lot. In other words, they keep on giving the same questions that were given like 100 years ago. They are still doing it. Like everybody's doing the same thing still. Things have evolved. Some of the time you have new questions, but all questions, old-fashioned way or techniques to do tests, they still have in this test. I don't know why, but they still have it. So you heard me saying all the time, you might not be seeing this test. Probably you're not going to perform this. Probably we're not doing this anymore. And you're going to be wondering, I know, because if somebody tells me something like that, I will be thinking the same. So then why are you teaching that? Because if you see it in the exam and I do not tell you, then you're going to say, oh, that teacher didn't say that. Oh, I didn't pass the exam because none of the questions were in my exam. Because if I'm going, move around with, technology, with the advances that we have accomplished up to now with technology in a laboratory, things are done totally differently. There are many things that are not done the way that they used to be done. Like, everything was manual. Most of the tests were doing manually over there. So can you imagine at this time, an era, and we're still doing all that? But unfortunately, the exam still giving you those questions. For that reason, we go over those. Even though if you're not going to encounter these tests, hopefully not. Because as far as I'm concerned, I don't recall seeing that anywhere that I'm being to, because, I've seen my students during the internships and they don't do, these medical doctors don't do it because they don't get paid for these tests. So if I don't get paid for this test and I have to spend so much time doing it, why should I do it? I just refer my patient to a laboratory. I just send the test, the specimen to the laboratory. Good in you? So that's basically what it is. So for the sake of the test, that's the reason we go over there. Now the test is done, but it's not done the way that, it says right here. It's done differently because now it's analyzed automatically using certain devices, certain equipment. So that's basically how it works. Okay, so you know that the degree of, or the angle that you have to use when you're doing a peripheral blood smear has to be 30 to 45. This is the angle. And the smear has to look like a feather edge. So that's basically what it is. So let's see if I have here one of the slides, because you're gonna look at it. You have an idea what it is, so you understand, and the person over there also explains the feather edge. I believe this is the one. Okay. Because when you see it, you kind of retain more than just someone telling you, how it's done. Because, we have different ways of learning, and one of them, the most common one, is that you show me, I see it, and I can do it, or I can remember a lot easier to do the procedure. Okay, so here it's smear. Pay attention.
Please make sure to always wear gloves when handling patient specimens. The view of a well-smeared, well-stained peripheral blood specimen is critical to the evaluation of patients with hematologic disorders. The equipment we'll need are 2 clean glass slides, preferably frosted so you'll be able to label them, a specimen of anticoagulated blood. This is usually provided for blood counts, and a lavender top tube which has EDTA, which will answer. Pasteur pipette with a bulb on the end of it. you will need to open the blood tube. You are going to place a small drop of blood on the end of the slide just in front of where the frosted area is. A little drop of blood about that size. It's perfect. Put the plaster pipette back in the tube. You are then going to take the spreader slide, which is going to be used to make the smear. Stabilize the base slide with your index finger, taking the spreader slide and holding it at about a 30 to 45 degree angle. You're going to bring it back, flush against the bottom slide, and when you reach the drop of blood, you'll see that the drop of blood will start to spread along the bottom of the slide. When it's spread to almost the entire width of the spreader slide, you are going to, in one steady, quick motion, keeping the spreader slide flush against the bottom slide, bring the spreader slide all the way to the end of the bottom slide. We're going to make the slide. The distal portion is known as the feather edge. You can see it's a much thinner area. You want to wait until the slide is completely dry. It takes about 5 minutes. And then after it's stained, the area of the slide that you want to look at to best judge blood morphology is just a bit proximal to the thin area of the feathered edge, right about here.
Okay, so that was it. We're not gonna go through the whole other process that is going to be doing, because this is actually not something that you're gonna be doing, but the purpose was for you to understand the angle at which the spreader is being placed on top of the slide when you're gonna be doing the peripheral blood smear, and the shape of the smear or how it looks like. Remember, feather edge. So that's basically what it is. We're gonna move on because that is something that I just want you to see and understand, even though if you don't have to do it, but for the purpose of the exam, remember. So that's basically what it is. Oops. Let's get this, erase this, too much stuff that I put in my slide. Well, I will erase them when finish. Okay, so gram stain, remember there are gram stain, positive and negative. Now, what is this important? Because sometimes you may have a question from here, not a big deal, but one of the things that you need to understand is when you're doing the gram stain, which is through the peripheral smear, but this is to detect bacterial infection. So there are two types of gram stain. There is a gram positive and gram negative. So how do we know it is positive? How do we know it's negative? Well, after processing this smear, they're gonna change colors. Remember, like the pregnancy test? They change colors, the little lines. If you don't see the second line, well, patient probably is not pregnant, or at least the test is showing that. So there is gram positive. If it is gram positive, the slide is gonna color purple. So stain means that, you know, that you're gonna see a color. So when you do the blood smear, if it turns purple, when you are processing, putting some chemicals on top of the blood smear, it's gonna turn purple. If it's purple, so you have a gram positive bacteria. That's basically what it is. And what is the gram positive bacteria? Staphylococcus. If it stains pink or red, gram is gonna be negative. And what is the negative bacteria? Streptococcus. So that's basically what it is. So gram positive, remember, is going to stain purple, and gram negative is going to stain pink or red. So here we have them. See, gram negative, pink or purple. And gram positive is going to be, once again, purple. I'm sorry, era purple for gram negative and gram positive. Oh, what am I saying here? Purple for gram positive. So I have it right here in the pictures, but I was memorizing this myself. So here we have gram negative, stain is pink or red. Gram positive is purple. So remember, this positive will be Staphylococcus. Staph. So that's basically what it is. And negative will be strep. Now, just because it says positive does not mean it's the bad one. It's the other way around. Gram negative is bad. Gram positive is good. Well, not that it's good, but it's the good bacteria. Remember, this is Staphylococcus, the good bacteria, the normal flora. Streptococcus is the bad bacteria. This stuff, we have it in our body, all over the place. It protects us. Streptococcus doesn't protect us. It's bad stuff. So we shouldn't have it. So that's basically what it is. So think about gram negative. Remember, pink or red. That's the color. So if you have a question, it will be based on that. Gram positive, it will be purple. Now, another possible question is, which is the normal flora? Gram positive or gram negative? Remember, negative does not imply good. Oh, my test was negative. That's not what we're talking about. This is the type of bacteria. Gram negative bacteria are really bad. This is the one that can kill us. Staph might not kill us because it's the normal flora. It's just that if it goes where it's not supposed to be inhabiting, then can cause problems. Anything else? No worries. So just think about gram negative. The stain is going to be pink or red. Gram positive, purple. Oh, even better. Positive, purple, P, purple. See, positive, P starts with the P, purple, P. So that's how you're going to recall a lot better this. The negative, you know already, that is pink or something else. So it doesn't match. So those are the things that, I give you as a clue, as a tip, so you can remember this when you're taking the exam. Cholesterol levels. Now, cholesterol levels, once again, this is chemistry. We are going to check for cholesterol levels. Now, cholesterol levels are very common tests done. So do not worry about all the information here. Just worry about what I'm going to tell you, because it's a lot easier. So we are going to check for cholesterol, and the only thing that we need to know is how do we get the cholesterol? Well, our body produces cholesterol, or we are ingesting the cholesterol through our diet, whatever we eat, right? So that's basically what it is. The cholesterol is needed in our body. It's just that has to be in between certain ranges. That's basically what it is. So don't pay attention too much about all this wording right here, because it's going to get you confused. You just know cholesterol levels. Now, high levels of cholesterol. What's the problem with cholesterol? Well, cholesterol block the blood vessels, particularly the arteries. That's called atherosclerosis. So atherosclerosis is nothing else than the narrowing of the blood vessels due to this fat inside the blood vessels. So that's basically what it is. And that can cause you heart attack or stroke. Now, let's talk about the types of cholesterol that we're going to have, because sometimes we just talk about cholesterol. But for the sake of the questions on your exam, you need to understand that there are 3 levels of cholesterol. It is not just cholesterol. Cholesterol is like when we say a mole, it's an umbrella, but under that umbrella, we're going to have other components of cholesterol. Think about blood. Blood is not just blood. What is blood? Plasma, red blood cells, white blood cells, platelets. So we have lots of things within. Here are the same. So we have a test that is called total cholesterol. What is total cholesterol? Total cholesterol is the amount of all the analysis of the cholesterols that we have, which is LDL, HDL, and triglycerides. That is the sum of all these is called total cholesterol. But we don't want to talk about just total cholesterol. I want to see the differential. Let's put it that way. Let's do the differential of cholesterol, which is all the components of cholesterol. So we have low density lipoprotein, which is LDL. This is the bad cholesterol. Pay attention to this because there are questions from here for the exam. So which is the bad cholesterol? So then you think about LDL, low density lipoprotein. Which one is the good cholesterol? HDL, high density lipoprotein. So usually they use the acronym LDL, HDL. So think about that, good cholesterol, HDL. Bad cholesterol, LDL. So that will be one of the questions. Now, the other question is then, which of the following tests requires fasting? What is fasting? Fasting is not eating anything 8 to 12 hours prior to performing the test. So if your doctor says, okay, so you got to be fasting to do this particular blood test, that means that they're going to tell you that you should not eat or drink anything with a period of 8 to 12 hours prior to the test. That's why these tests are ordered early in the morning. So during the night, you don't eat, you don't drink. So you go to the clinic, you go to the laboratory, get your test done, then you go for your breakfast. Don't eat or drink anything prior. That is fasting. So cholesterol is one of those tests that needs fasting. It is required to do it. What is another test that is required to have fasting? Glucose. Glucose levels, you need to be fasting. So these 2 tests, try to remember because I'm gonna remind you again when we get to phlebotomy. But remember, what is total cholesterol? LDL, HDL, and triglycerides. So if you have in your questions, which of the following tests requires fasting? Then if you see LDL, that's the answer. If you see HDL, that's the answer. If you see triglycerides, that's the answer. If you see cholesterol, that is an answer as well. We are talking about for the lipids. That's basically what it is for the cholesterol levels. Now it's going to be another question for glucose, then fasting for glucose. And for how long you're going to fast? 8 to 12 hours. We will discuss that again when we get to phlebotomy. But for now, think about this because that is how the test is done. They can give you just cholesterol. Oh, cholesterol, I know is fasting. But if they give you LDL or HDL or triglycerides, and you're not familiar with these terms, you're going to get the wrong answer because glucose is not going to be part of it. Because otherwise glucose will be, but they are talking about cholesterol. So don't forget that can be 1, 2, 3, 4 options right here. Cholesterol, LDL, or HDL or triglycerides. See? 1, 2, 3. Oh, but by the way, it's 4. So 4 options. So that's basically what it is. So keep it in mind, write it down, put a note right there, because you definitely will have these questions on your national exam. Let's talk about cholesterol levels. This is another question for exam. I don't know, I didn't highlight it. That's not good. I highlighted right here. This is it. What is the normal cholesterol levels? Because they're not gonna write down desirable. They're gonna write down what is the normal cholesterol levels. Less than 200 milligrams per deciliters. That's it. That is the answer. You should not have more than 200 milligrams per deciliters of cholesterol. So just remember 200, less than 200. It's not equal to 200, it's less than 200. Now, anything else, no worries. What is borderline cholesterol? 200 to 239 milligrams per deciliters. What is it that you've been diagnosed already with cholesterol? Because borderline means you are there, you are close to develop cholesterol. So if I put you in a diet, if I ask you to do exercises, if I ask you not to smoke, not to drink, well, your cholesterol levels are going to go back to normal. But if you don't pay attention to that, they enroll your diet, you don't do exercise, and you keep on eating whatever you want, then your cholesterol levels are going to go above 239. So anyone who has over 240 milligrams per deciliters of cholesterol, you have high cholesterol. Now I have to medicate you. So that's basically what it is. But the question for your exam is going to be, what is the normal ranges, or what is the normal value for cholesterol levels? Less than 200 milligrams per deciliters. Any questions? So don't forget about this, because believe me, you're going to see this question. And these questions are straightforward. They're not going to ask you about borderline, high cholesterol. No, no, no. They're going to ask you about, fasting, which test you're going to fast, particularly when it's based to cholesterol and glucose as well. But this will be just for this part of the chapter. So when you're going to do the cholesterol levels, you're going to do something similar of what we did with, hematocrit, hemoglobin. So we're going to get a sample of blood, and then you're going to use a particular device or machine to do this. So that's basically how it is. So it's very similar to the HemoCute when we did the hemoglobin. So these tests are not done manually. This test requires this. The only test that requires us to do it manually is the dipstick, the reagent strip. That's when you can dip it inside the urine and kind of, compare with the bottle, model the chart, and then write down the results. The other ones, all of them are going to use, once again, a analyzer. It can be automatic or semi-automatic. Automatic means that you just put the sample inside the machine and the machine read it for you. Semi-automatic means that you're going to place it, then you're going to press a button to start, and then to print it and all that stuff. So you do a little something manually, but nothing else. So that's basically how it works. Remember, if we use a finger stick to do this, because it's very similar to hemoglobin, we're going to do it from the finger, because you need 1 drop. And then once again, we're gonna wash our hands. We're gonna wear gloves, and we're gonna clean the finger. Remember, 2 fingers only, 3rd and 4th finger, middle and annular finger. So remember that, because those are questions for exam. Then you're gonna clean it. You're gonna let the finger air dry. If you have noticed it in these videos that I've shown you, somebody with the gauze comes and wipe it off, clean or dry the finger. No, you should not be drying it off with anything. You let it dry. Then you prepare your equipment. Then you do the stick, and then you're gonna wipe the first drop. It's only one drop, not three, not two. Once again, this is the new technology that was done back in the days. There are many things that have not changed. Some of the things with the videos have not changed, but the new these are new questions. So those are new. Some others stays the same way as before. So once again, you're gonna kind of time it, because remember, when you place the drop in this cassette and then you place it in the device, I'm not gonna mention names of devices because it can be any brand. I don't know which one you're gonna do, but it is a cholesterol device analyzer. Now you're gonna place your, you're gonna check your timer or your watch or your clock. You're gonna be looking at it, whatever the case may be. Then you're gonna read the results. Then you're gonna document them. So that's basically what it is. We're not gonna go video or anything like that with this because it is very similar to hemoglobin. Not much for you to do except for the finger stick, placing, the cassette right there to kind of retract or absorb or suction the blood from the drop of the finger, and then make sure that, it is well done. So that's basically what it is. Here it is another test that I was talking about before, glucose levels monitoring. So remember, sometimes we have the patient, we do glucose in a regular chemistry test. So we send it to the laboratory, and all the tests for chemistry is gonna reveal whether your glucose is high or low. But sometimes we do these independent tests. Remember, sometimes in the office, at the POL laboratory or the physician's office laboratory, we're going to run some things that are CLIA approved. That is CLIA wave tests, which I mentioned before. Don't forget that, because that's very important, because these are the tests that we're going to run in the doctor's office. These wave tests are not done in a laboratory because they have all these analyzer machines. This is for the medical doctor that doesn't have this big equipment and professionals to analyze and read and give you the results. This has got to be a lab professional. Has to be, remember, a microbiologist, has to be a chemist, doctor, it has to be a pathologist or hematologist or anyone else. In our doctor's office, we don't have any of those. We don't have specialized people to do these tests. So we run tests that are low profile. That's basically what it is. So remember, it says right here, fasting blood sugar. Let's think about cholesterol as well. It says right there, no eating or drinking for 12 hours. It's 8 to 12 hours. So remember, 8 to 12 hours. That's the timing. We're always going to have ranges in what's considered normal. Now it says right here, except water. All depends on the test that you're going to do. So for the glucose, you might be able to drink water because water doesn't have any complements within, doesn't have any ingredients, so to speak. Water is water, doesn't have anything that can change the amount of glucose that you have there, meaning can add sugar to the blood, because water doesn't have any components. So that's water, that's it. So that's basically what it is. So the normal ranges of glucose are in between 70 to 110 milligrams per deciliter. This is the normal. So someone who has more than 110 is not diabetic. It has to be over 100, over 120 or 24. So that basically depends on the laboratory that is evaluating, analyzing this blood. But normal ranges of glucose in anyone has to be between 70 to 110. So that's basically what it is. If we suspect that someone might be having high levels of glucose, because we already the chemistry test, the regular, the routine test, where it checks cholesterol, sodium, potassium, magnesium, blah, 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 blah. So then it comes the results and it says, oh, your glucose is high. But that test we did it randomly. Remember, the patient came over to the office without fasting and we did the test. Now, probably was because the patient ate something very sweet prior to the exam. Now we're going to do a fasting to make sure that we actually following all the protocols to make sure that the levels of glucose are exactly the levels that you are having in your blood, not something that you ate that up to the count. So that's basically what it is. So pay attention to this part, like fasting. We're going to go over phlebotomy. Remember, we're going to talk about this again. We're not going to talk about the normal levels or ranges of glucose, but the normal levels, remember, 70 to 110 milligrams per deciliters. Now, this is another question for exams. Postprandial. So let's highlight this because I don't want to highlight the whole thing, just this part right here, postprandial test. So when the patient is already being screened for diabetes, meaning you ran the first test, it came high, now we ordered a fasting glucose test, and now it's high again. Now we're gonna order 1 hour, 2 hours, 3 hours, 5 hours glucose tolerant test. That's how it is. The medical doctor is going to order this test. The patient is going to drink a very concentrated, sweet stuff, and we're going to run the test after that. So that's basically what it is. Now, we're not going to talk about directly the test just now. We're going to talk about how it's done, because this test has to be done 2 hours prior. So postprandial. Remember, post, after. Prandial, meals. That's what it means. So every single time that you have a test, glucose tolerant test, is because you're gonna do the test after you have been taking this sweet stuff. So that's basically what it is. So they're gonna give you, okay, what is postprandial means? Then you look for a question that says that 2 hours, 3 hours after meals, but the answer is 2 hours after meals. This is the test that is done 2 hours after meals. That's called postprandial. That's basically what it is. So keep it in mind. We're gonna talk about this again during phlebotomy, but for now, just think about this. When someone is diabetic, or we suspect that this person is diabetic because we have run 2 tests already and the glucose levels are above 110, we did already the fasting, is still over that amount, we're going to do the postprandial test, which is the glucose tolerant test. Remember, glucose tolerance test means you're going to see how much you can tolerate your glucose. So when you drink this very sweet stuff, they are going to do the test and they're going to see how fast this very sweet stuff that you drank is going to be removed by the blood, and it's not going to stay there. But if it stays, we know already that you have diabetes, and that's when we start treating you with medications. So remember, post means after. Prandial means meal, so after meals. So there is a glucose tolerant test that is done after meals. So remember that. That means after meals. Here it is, glucose tolerant test is a very sensitive to the ability to metabolize glucose. These terms are very, very difficult to understand. So for that reason, I told you that the person is being given this particular sweet stuff, very sweet. That is disgusting. I kind of tasted, want to see why my patients were vomiting after I gave this, or what is it that they didn't want to continue drinking it, because it's very, very sweet. So again, this is given also for females who are probably suspected or having gestational diabetes. When someone is pregnant, you know, and you suspect that this person might be diabetic, or you know, the results are higher on glucose, we're gonna give this to the person, to the pregnant females. So remember, it's to detect diabetes. It doesn't matter what type of diabetes it is. So once again, this test is collected, and it can be done 1 hour, 2 hours, 3 hours. That's how it's done. So meaning that after I give you this very sweet stuff, I do a test 30 minutes later, then 1 hour later, then 2 hours later, and then 3 hours later, or 5 hours later. So that's how it is. So all depends, and you have to be fasting because you've been given this to determine if you have diabetes or not. So when you see this glucose tolerant test, or GTT, first of all, think about fasting. Second of all, that this patient is going to be postprandial, which means after meals. Remember, postprandial means after meals. So that's how you need to understand this. Any question about these glucose tests? Remember, most of the time, glucose test is to determine whether you are diabetic or not. But in a regular test, in a routine test, you're not complaining about anything. You just went for a test. Well, we're gonna just check chemistry glucose, no fasting. Then if your results are higher than normal, more than 110 milligrams per deciliter, then we're gonna order fasting glucose test. If that test comes elevated, now we're gonna do glucose tolerant test. So this is kind of a process. This is not just one test. Oh, you run one test, oh yeah, it's this number. And it's not any number. You have to run at least three tests in different times. I do a test today, fasting, a test a week later, fasting, and another 2 weeks later, fasting. And all these tests comes higher than 124 milligrams per deciliter. Now you're diabetic. Just having more than 110 does not mean diabetic. You are hyperglycemic. Your glucose is higher, but not diabetic. So you have to meet certain requirements for us to consider you diabetic. So that's basically what it is. Just for you to understand. Some people think, oh yeah, this is done this way, so I have diabetes. Oh, your glucose is high, diabetes. No. Once again, that is the reason why you should not give results to the patient, even though if you see the values, because it's not, something that you should do, number one, and it's not that definite. This test has variations. The patient is not diabetic. Who say that the patient is diabetic? It's not diabetic. You have to run all these tests, and at the end, the medical doctor that proves that all these 3 tests in different weeks, fasting, give the number or the glucose tolerance test that you have done, 1 hour, 2 hours, 3 hours, 5 hours, all of them came positive, now you're diabetic. You don't diagnose someone by getting one value. You don't diagnose hypertension because the patient came to the office and you check the blood pressure and the blood pressure is high. Oh, you have hypertension. No, that's false. That's not how it works. So for that reason, you should not tell the patient results, remember. Because this patient is going to go to the medical doctor after you check the result, if you do something, and it's going to tell, oh, the nurse told me that I have diabetes. They told me that I have hypertension. They told me that I have high levels of cholesterol. No, you don't say anything until the medical doctor diagnosed the patient and run all these tests. This is not as simple as that. Oh, well, yeah, I'm poor and rich. Yeah, I have a lot of money. I don't have. That's not that way. These labs can be false positive, false negative. They were not done correctly. The tubes were expired. The laboratory made a mistake. There are a lot of things that can play a role in determining the values of. That's why we repeat the test different times in different occasions, with you having different procedures prior to the test. So that is how it works. So for the glucose tolerance test, we're going to do exactly what we did for hemoglobin, because for cholesterol, we're just going to use a test strip, a little very tiny strip, and we're just going to, place it close to the drop of blood from the finger, from wherever else, and that's exactly, and then we place it in the glucometer and do the test. This is a very, very common test. You might have to do it. So for that reason, when we get to phlebotomy, I'm going to show you how that works. We actually going to do the glucose test, not the glucose tolerance, but just the regular glucose test using the glucometer. So that's basically how it works. Very simple test. So we're going to see it. Probably some of you know how to do it. You see how it's done, because we have tons of diabetic people. So you just need to do it correctly. Remember, the first drop of blood needs to be wiped out. You do not use that. We always have to document every single results if that's the case. So again, or we give the results to the medical doctor. Listen, these are the results. But you see it on the screen or the monitor where you're doing the procedure. So you just do it. Once again, very simple. Just hold on a minute. We're gonna just watch a short videos, because all these are very short videos that I want you to see for you to have an idea if you have never seen something like this. Some of you probably are familiar because, once again, diabetes is very common. Okay, so here it is. Pay attention.
We're going to do a finger stick. We'll be doing a glucose check. Okay, so we got our supplies here. We got our glucose strips, our glucometer, alcohol bag, gauze, Band-Aid, and Lancet. This is the little needle, small needle that we're going to use to do her finger stick. Okay. So of course we know that we're going to use either the middle finger or the ring finger. In other words, we're going to use the third or fourth finger. Okay. We want to avoid the pinky, the index, and the thumb. Okay. So what I'm going to do is I'm going to go ahead and clean her first, and while the alcohol is drying, I'm going to set everything else up. So we're going to clean her here, wipe it from the inside out while that's drying. Did you want to do that finger, or was it the middle finger? Oh, you want to, oh, okay, I'm sorry. Sorry about that. Like I have more. Yeah, most people go for the middle finger. I usually go for the middle finger too when I have to get a finger stick.
Because it's larger.
So, one thing I want to mention, we're not on top of her finger, we're on the sides, okay? All right, so I'm gonna clean that, let that dry. While that's drying, I'm gonna go ahead and open the Band-Aid. Put my dirty stuff over there to the side, open up my Band-Aid. Now, with these strips and the glucometer, to turn it on, all you have to do is enter the strip. And you don't want to enter the strip until you're about to stick, because if you enter it too soon, the machine is gonna start hibernating or going to sleep. So you only want to enter the strip once you're about to stick, and the machine will let you know when it's ready for the patient's drop of blood, okay? All right, so we had to get a new glucometer and new strips because that machine was giving us an error message, but same thing. We're going to enter the strip in, and that's what's going to turn it on. So we don't press a button or anything like that. We just enter the strip, okay? And it's going to turn on, and it'll let us know when it's ready for the blood. You'll see a drop of blood there. That's how we know this is ready for the blood, okay? So usually what I do, I grab a piece of gauze, okay? Because we do have to wipe away that first drop of blood on the patient's finger. It's ready for the drop of blood, and this is where we're going to place the patient's blood once we stick her finger. So we're going to take the tip off the lancet. Now, this lancet will have to press on the patient's finger, hit the button. Some lancets, you just press it down on the patient's finger and a needle will spring out. But in this case, we do have to hit the button. So what I'm doing here, I'm just kind of milking her finger just a little. And notice I'm here on the side. I'm not on the top of her finger. I'm right here on the lateral side, right there, on the lateral part of her finger. Alrighty, so I'm gonna just milk it just a little. And then I'm just gonna stick, okay? And we gotta wipe away the first drop of blood, right? So I'm gonna wipe that away. And then with this machine, all you have to do is allow the blood to just, here we go. And it'll take a